Welcome back. Today's case is that of our fictional patient named Arthur, a 60-year-old architect who comes to see you, his doctor, after feeling tired for about 3 months. He has no past medical history, he takes no medications, his vital signs are normal, and physical exam is normal. So you decide to draw some blood. Let's look at his blood first before we read his lab report. We put a drop of his blood onto a glass slide, smear it, and stain it. In hematology, we can use 2 different dyes. The first is eosin. It's an acidic stain, so it stains basic things like proteins reddish pink. The other is methylene blue. Methylene blue is a basic stain which stains acidic things like nucleic acids, like the DNA in a nucleus, a deep blue to purple color. After staining Arthur's blood, here's what the slide looks like. If you have no idea what you're looking at, no worries. We will study some normal blood first before we study Arthur's blood to answer this question. What is the most likely diagnosis? You can pause here to decide if you'd like. A normal peripheral blood smear has lots of red blood cells. Red blood cells in the blood do not have a nucleus. Instead, they're mostly made up of hemoglobin. And hemoglobin is a slightly basic protein that eosin, an acidic dye, will stain. Between the red blood cells, we have these tiny cell fragments here called platelets. They have a role in coagulation or blood clotting. They come from the megakaryocyte in the bone marrow. So little bits and pieces of this megakaryocyte get ripped off and become platelets. Also scattered around are white blood cells. We have different types of white blood cells. First up is a segmented neutrophil, which has a basophilic nucleus. All of that DNA in the nucleus loves and is picking up the basic stain, hence basophilic. A neutrophil has a single nucleus that's segmented. It can almost look like some sausage links. And in the cytoplasm, there are granules, but they aren't bright red, so they're not eosinophilic. But they also aren't deep blue, so they're not basophilic. Rather, these granules appear neutral, a lilac color, and that is how they got their name of neutrophil. A slightly younger neutrophil is a band neutrophil here. Next up is a very distinctive cell with its basophilic nucleus and prominent reddish eosinophilic granules. This is an eosinophil. It also has an interesting nucleus. It is a single nucleus that is bilobe. This cell over here has basophilic granules. It is a basophil. The names make sense, right? Eosinophil, basophil, neutrophil. Basophils can also have a bilobe nucleus that is hiding behind these prominent basophilic granules. Our fourth type of cell is the one accompanying our neutrophil here. This is a monocyte. This is a huge cell. It is the largest of the white blood cells. It hangs out in the blood as a monocyte, but if it's signaled to leave the blood and enter the tissue, it becomes known as a macrophage. Macro meaning big, phage meaning eater. This is a big eater of things, and it eats things like microorganisms and cellular debris. And last but not least, our lymphocyte with its classic round nucleus inside a round cell. Here it is. The nucleus is about the size of a normal red blood cell, and then it has this tiny rim of cytoplasm around it. Back to Arthur's blood. Compared to a normal blood smear, Arthur's blood looks very busy. It has a lot more nucleated cells than normal. So our first job is identifying the cell. Is it a myeloid cell? The myeloid family includes our 3 granulocytes, which are neutrophils, eosinophils, and basophils, plus monocytes, or is it lymphoid? Arthur's cells in question here are lymphocytes, so we're talking about the lymphoid family. He has way too many lymphocytes in his blood. Take a look at his lab report here. He has 60,000 white blood cells per cubic millimeter. That is way too much, way too many white blood cells or leukocytes. So there is a leukocytosis, and that leukocytosis is caused by a lymphocytosis. You can see here that 85% of the white blood cells are lymphocytes, which is what we saw in our smear. His hemoglobin and platelets are okay. Going back to our answer choices, we can knock out chronic myeloid leukemia or CML. CML would look like this. Another busy slide, but the busyness is due to the cells of the myeloid family. You can spot some eosinophils here. There's some neutrophils. We also see some cells that we haven't met yet, but they don't belong in the peripheral blood. Our next option is chronic lymphocytic leukemia or CLL. We can keep that as an option since we do have lymphocytes. What about hairy cell leukemia? Hairy cell leukemia would present with much lower numbers. The cell counts are lower across the board. There is pancytopenia, so these lab values do not fit Arthur's case. Plus, there's usually splenomegaly in hairy cell, which Arthur didn't have, And lastly, the lymphocytes of hairy cell leukemia look a little different. They can look slightly larger with these projections that form around the cell. They almost look like little hairs that come out of the cytoplasm, hence the name hairy cell leukemia. What about aplastic anemia? Again, the labs would be different from what we saw in Arthur. We would also see a pancytopenic picture, but the numbers would be even lower than that of someone with hairy cell leukemia because the bone marrow has very little hematopoietic cells around to make the cells of our blood. And finally, is it an acute leukemia? No. If it was an acute leukemia of the lymphoid family, we would see lymphoblasts in the blood. Here is an example. If we grabbed one of Arthur's lymphocytes and placed it next door to this lymphoblast here, you can see the difference. If it was an acute myeloid leukemia, we would see myeloblasts like this one here. And now check out the comparison to Arthur's transplanted lymphocyte. Arthur has CLL, where his blood has way too many B lymphocytes in it, and these are not normal B lymphocytes. These are atypical, neoplastic, malignant B lymphocytes. Thank you for watching. I hope you enjoyed this quick case. If you did, please hit the like button and subscribe to my channel for upcoming cases. Let me know what you think about these case-based videos and comment below on any requests you may have.